All right, so we are recording now. We've talked about the structure of DNA and the way in which the DNA can be replicated. But to get much deeper into what it actually does, I've got to get into a second class of large biomolecule known as proteins. Okay, there we go. So Watson and Crick worked out that DNA is shaped like this double helix. The reason that matters is that being shaped like a double helix allows DNA to be duplicated through what's sometimes technically called semi-conservative replication, which means that when a piece of DNA duplicates, each strand separates from the other strand. The two separate strands move apart, and then each separate strand can be used to build a complementary strand. And we talked about that in the last lecture. DNA is, of course, a polymer of these subunits called nucleotides. To be more accurate, it's two polymers, two strands of nucleotides wrapped around each other, held together by hydrogen bonds between the bases. And each nucleotide, again, consists of a phosphate group, a sugar, and one of four possible bases: adenine, thymine, guanine, and cytosine. What's critically important about DNA is the order that the nucleotides are in. We'll talk a bit later as to exactly how they do this, but it's possible. It was worked out in the 80s, and it's gotten a lot faster and more efficient since then. It's possible to take a DNA sample and literally read the order that the nucleotides come in on one of the strands. In fact, we do this quite a lot in everything from evolutionary studies to forensics. When you hear about people doing DNA testing, this is one of the ways in which it can be done. You can take a DNA sample and read the order of nucleotides, and the order of nucleotides is what's critical. It was hypothesized early that DNA might just be a repeat ATGC, ATGC, ATGC on one strand, and so on. It turns out it's not. This is an actual sequence of human DNA. Somewhere in your cells, you've got a piece of a chromosome where one of the DNA strands goes CAG, CT, GGG, GGT, AA, GGG, GGG, CGG, ATT, ATT, CAT, AT, AATG. I hope you're writing all that down. Actually, I'm kidding. Please don't write that down. But what's important is the order that those nucleotides are in. That order means something. To get into what that order means, I have to start talking about a second class of biological molecule. That's the proteins. You did some testing for proteins in that foods lab a couple of weeks ago, but what are they? Well, a protein is a polymer of amino acids. Just as DNA is a polymer of nucleotides, and PVC is a polymer of vinyl chloride monomers, and cellulose is a polymer of sugars, proteins are polymers of amino acids. And there are thousands of possible amino acids, but only 20 of them are used to make up proteins. Proteins do an absolutely ridiculous number of things in living systems. The filaments in your muscle cells that make them contract are made of proteins. The hemoglobin in your blood that carries oxygen from your lungs to the rest of you is a protein. The insulin that tells your cells to start absorbing nutrients from the food you've just digested, that's a protein. the stuff that makes up your hair and your fingernails, that's a protein. There's a stonking crazy number of proteins that every living cell has to make. Each protein's got a different shape that makes it possible for it to do what it does. If you had hemoglobin proteins of the wrong shape, your blood would not be able to carry oxygen, or at least not carry it efficiently. The shape is determined by the order that you put the amino acids in. Just as the meaning of a piece of text is determined by the order you put the letters in. The order that you arrange the amino acids in determines the protein shape. And what's critical about this part of the course that you get is that the shape of the protein determines what the protein can do. Just as the shape of a hammerhead or screwdriver head or socket wrench socket determines what that tool does. And that order of nucleotides in DNA essentially tells a cell what order to put the amino acids in to make a particular protein. That's the information that DNA carries. It carries information in the order of nucleotides. How it does that, we're going to look at later this week. But let's backtrack and see what an amino acid is. Just as a nucleotide is made of three parts, a sugar, a phosphate, and a base, an amino acid consists of three major parts. There's a nitrogen with a couple of hydrogens stuck onto it that's called the amino group. There's a carbon with two oxygens stuck onto it. That's called a carboxyl group. Incidentally, there's a central carbon right here and a little hydrogen that's kind of tagging along for the ride. But the third major part that's attached to that central carbon is called the R group. And different amino acids are defined by having different R groups. They've all got aminos. They've all got carboxyls. What makes one amino acid different from the other is that they'll have different R groups. And that's why it's important for pirates to eat a lot of amino acids, because they need to have those R groups. That was a joke. I'm imagining you all laughing uproariously right now. As I've said, there's thousands of amino acids. Only 20 of those are used to assemble proteins. There are other amino acids that play other roles in living systems, but that aren't used to assemble proteins. One that some of you might have heard of is ornithine. It's not used to make proteins. It's got other roles in metabolism, and it's actually sold as a dietary supplement. I couldn't tell you exactly what it does, and I don't actually know if those supplements work, so this is not intended to be an ad for them. But this is one amino acid that you might have heard of that is used in living cells, but is not actually one of the 20 that's used to assemble proteins. Of those 20, 9 of them have to be present in your diet. There's no way for human cells to make them. There's actually a 10th that is necessary for infants, but adults are not so dependent on it. But yeah, for adults, there's 9, and we call those essential amino acids. And you might have heard of those if you're in the nutrition program, or if you've read much about nutrition on your own. The other 11 your cells can make if they don't get them, but there's 9 that you can't do without. And food that provides enough of all of these 9 is said to be a complete protein. So a number of cultures might live primarily on rice and beans. That's actually a complete protein. You can get enough of all 9 essential amino acids on a diet based mostly on beans, rice, and corn. If you don't get enough of one or more of the essential 9, you can end up with various deficiency diseases. So that's another context in which you might have heard of amino acids. So again, an amino acid has an amino group, which is a nitrogen with a couple of hydrogens stuck on, a carboxyl group, which is a carbon with a couple of oxygens, and an R group. And the R group can be as simple as a single hydrogen atom in the case of the amino acid called glycine, or the R group can be a long chain of carbons, nitrogens, and hydrogens, a really big R group, as in the case of this one, which I forget, I think it might be valine. Valine, I could be wrong about that, but I think that's what it is. Do not try to retain any of this. This is just illustrations. These are 8 of the 20 main amino acids. Okay, that's not alanine because alanine only has just one carbon and a few hydrogens as its R group. Tryptophan, which allegedly makes you sleepy after Thanksgiving dinner, although I think that's not actually true. The R group is a double ring of carbons mostly. Phenylalanine has a single ring of carbons. Methionine's got a little sulfur in it. That, by the way, is why there's that S in schnapps. A couple of critical amino acids contain sulfur, and that's why you need it. Would we need to know what groups they are in? Don't worry about the groupings of amino acids. You do need to know that each amino acid contains an amino group and a carboxyl group and an R group. Don't worry about classifying the amino acids into polar, nonpolar, or whatever they are. Here's some more. This is another one with some sulfur in it. Glutamine's got a branching R group, and tyrosine's got a carbon ring. And there's yet more. Okay, arginine was the one with the great big R group that I showed you right there. And again, do not try to memorize this. This just gives you an idea of what essential amino acids look like. All right. 2 amino acids can form bonds between the amino group of one amino acid and the carboxyl group of another. And this is what lets you string amino acids together in polymers that can be as long as 1,000 amino acids. And the amino of each one bonds with the carboxyl of the next. So they're bonded amino to carboxyl, like the nucleotides in DNA are bound phosphate to sugar to phosphate to sugar to phosphate to sugar. As an example you might have heard of, on the left, you've got an amino acid called aspartic acid. There's the amino. There's the carboxyl group. The amino is a nitrogen with a few hydrogens. The carboxyl is a carbon with a couple of oxygens. And this would be the R group up here. And then phenylalanine over here, there's the amino. There's the carboxyl group. And then this big ring of carbons and hydrogens is the R group. That's phenylalanine. When the carboxyl group over here forms a chemical bond with the amino group right here, we call that type of bond a peptide bond. Ooh, look, computer animation. Here, let's do that again. There's the separate amino acids, and a chemical bond can form between the carbon on the carboxyl group and the nitrogen on the amino group of the next amino acid. And we call that Dun, 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 a peptide bond. And by the way, a molecule of water as created as a byproduct. And so in green there, that shows you the peptide bond. Incidentally, if you stick an aspartic acid to a phenylalanine and then you tack on a few atoms to the carboxyl of the phenylalanine, you have a compound called aspartame, which is otherwise known as NutraSweet. Okay, let me go back one because someone's asked me to explain the peptide bond. A peptide bond is a chemical bond that forms between the carboxyl on one amino acid and the amino group on the next amino acid. So it's a bond that would form between this carbon right there and that nitrogen right there. This group of a hydrogen and an oxygen would break off. This hydrogen right here would break off, and those would go on and form a molecule of water. And what we're left with is a chemical bond. And again, we don't really need to know how these bonds work for our purposes in this course. Just know that atoms can be made to stick together. And that sticking together is what we call a bond. And the specific bond between a carboxyl carbon and an amino nitrogen that links two amino acids together is what we call the peptide bond. And then if we had more amino acids, this carboxyl group over here could form a new peptide bond with the amino of some other amino acid. And so we can put amino acids together in chains where the carboxyl of each one forms a peptide bond with the next. And we could link amino acids together in chains of arbitrary length. Some proteins contain over 1,000 amino acids, one after the other. Just as you can link up cars in a train by putting the linchpin on one car into the latch on the next car, and just keep doing that over and over until you've got a train of as many cars as you like. You can do the same thing with amino acids. All right, hopefully that helps. And yeah, if you stick a few more atoms on the end right here, you'd have a compound called aspartame, aka NutraSweet. NutraSweet is basically two amino acids stuck together with one peptide bond. But you could add more amino acids on the end and make a chain as long as you cared to. So we can describe the structure of a protein on 4 different levels. We can talk about the order in which you put amino acids together, and we call that the primary structure. So any single protein works because it's made of amino acids that are put together in a specified order, and that order is the primary structure. As soon as you make a chain of amino acids, the amino acid chain begins to fold. And the folding is caused by attraction between atoms that have an electrical charge on them. And when parts of that amino acid chain start folding, we have secondary structure. There's another level of folding that's caused by electrical attraction and repulsion between different amino acids R groups, and we call that tertiary structure. And then finally, if you have several amino acid chains that fit together into a single molecule, we call that quaternary structure. And you need to know all 4. So let me take you through what these look like. This is a computer model of what a simple polymer of amino acids would look like. Basically, each atom here is being represented by a stick of a different color. This blue thing right here is an amino group, and and that's a carboxyl, and the carboxyl is bonded to an amino, and there's a carboxyl bonded to an amino, and there's a carboxyl bonded to an amino, and a carboxyl group bonded to an amino group, and so on. This is just one amino acid after another, all of them strung together like cars in a train or beads in a necklace. Incidentally, a polymer of amino acids is sometimes called a polypeptide, or sometimes just a peptide for short. A protein might consist of one polypeptide, but it sometimes consists of more than one, and I'll talk about that in a bit. But here, for example, is the primary structure of the human protein insulin. First, at the beginning of it, there's an amino acid called methionine. That forms a peptide bond with the amino acid alanine, which forms a bond with leucine, which forms a bond with tryptophan, which forms a bond with another methionine, which forms a bond with arginine, which forms a bond with leucine, then proline, then leucine, then alanine, then leucine, then alanine, the leucine, the tryptophan, the glycine, the proline. Okay, I'm not going to read the whole thing. The reason why this all matters is that if you can't make insulin, or if you make insulin with a faulty structure, if you make faulty insulin, you have diabetes, or one form of it anyway. What's vital for your health is you have to have the ability to make insulin of the right type. If you can't make insulin that functions right, then you've got a health problem. You've got severe diabetes if you can't make insulin correctly. For you to make the right type of insulin, you have to be able to put amino acids together in the right order. If the cells in an organ called your pancreas cannot put amino acids together in exactly this order, then they won't make insulin that has the right shape, and that insulin won't work, and you'll be severely ill. You have to be able to assemble amino acids in the right order in order for the protein that you make to have the right shape to do whatever it is that it's supposed to do. Boring, but that's why this matters. When atoms in amino and carboxyl groups that are close to each other attract each other with an attraction that you can think of as being analogous to static electricity, the chain of amino acids starts bending and folding. And what will happen is that a part of a chain of amino acids may kink up into a type of spiral. And the name for a spiral of a chain of amino acids is an alpha helix. Not the same thing as the double helix. Double helixes are what DNA makes. Alpha helices are what chains of amino acids sometimes make. And it's caused by attraction and repulsion between atoms in amino and carboxyl groups that are fairly close. So no sooner do you string a bunch of amino acids together, then parts of that string will kink up and make this spiral structure, kind of like a telephone cord, if any of you remember what those even were. Okay, like a bed spring or a slinky. Alternately, when a chain of amino acids forms, sometimes the chain will fold back and forth and make a structure that's called a beta sheet. Instead of making a coil, that's an alpha helix, the chain of amino acids might do this kind of zigzaggy thing and create a structure that's known as a beta sheet. Sometimes it's called a beta pleated sheet because most of the atoms fall on a surface that looks a little bit like a folded sheet of of paper if you're on drugs. So I'm kind of doodling right there, but that's what I was doodling was a chain of amino acids. Can I go back when I have time? Where do you want me to go back? Back to secondary structure, to the alpha helix. Okay. Secondary structure is what you get when atoms and amino acids that are close together in the chain begin sticking to each other. A little bit like what happens when you have to try to wrap a package and the tape starts sticking to itself. That's certainly annoying when you're wrapping a package, but it's vital for proteins to develop the right shape. Alpha helices are what you get when a chain of amino acids will fold up into a spiral shape. They'll make a spiral that looks a little bit like a spring. And again, this is not the same thing as the double helix. Double helix is what DNA does. Alpha helices are what proteins make. Okay, can I go on? Okay, so secondary structure, parts of a chain of amino acids will either make a coil, an alpha helix, or they'll make a kind of zigzag that we call a beta sheet. This is a diagram of what it looks like. Those dashed lines that you see right there are hydrogen bonds, this weak bond type that we've already talked about, forming between carboxyl and amino groups. So that's a beta sheet. In tertiary structure, R groups can attract or repel each other because atoms have electrical charges on them. So this is a little bit like the way static electricity, the way that, a comb that's been drawn through your hair might attract or repel little bits of paper, or might attract or repel your hairs. That's static. In tertiary structure, atoms in the R groups cause the entire chain, not just parts of it, to fold up. This is a diagram of a protein that's found in your muscles. It's critical for getting oxygen to your muscles so that they can contract. It's called myoglobin. And instead of drawing individual atoms, we're just drawing the chain of amino acids kind of like a wire, instead of trying to show every little atom. And myoglobin happens to consist of several alpha helices. They are the parts of the amino acid that are drawn as spirals, but the entire protein is folded. Instead of just being a single chain with a bunch of spirals in it, it's folded in on itself. That's tertiary structure. And myoglobin would not work, and your muscles would not get the oxygen that they need if you could not put together amino acids in an order such that they kink up into a bunch of alpha helices, and then the whole thing crumples into this exact shape. That's a tertiary structure of a protein from cells in the intestine. Looks kind of like a one long ribbon. That's because this particular protein has formed a lot of beta sheets, and that's secondary structure. The tertiary structure is the way that all of these beta sheets have wrapped around each other. And then finally, this is a protein that's important in digestion. In this one, the alpha helices have been shown in red, the beta sheets are in green, and the parts that are neither are shown in purple. And this looks like a horrible mess, I'm well aware. What you need to know is that if the chain of amino acids that makes up triosisomerase does not first fold into alpha helices and beta sheets, and second, the whole thing does not fold up into this shape, triose isomerase is not going to work. Beta sheets are what you get when a chain of amino acids starts forming a kind of zigzag shape. And if you diagram what they look like atom by atom, they would look sort of like this. That chain of amino acids is kind of going out this way and then coming back and then coming back again. And creating zigzags like that. Okay, and then the alpha helices and beta sheets can further wrap around each other, and you get what we call tertiary structure. And these are a couple of more examples. These are all computer graphics. Alpha helices are shown in blue, beta sheets are shown in red, regions that are neither are shown in gray. And again, this is not, you don't have to memorize this kind of detail. You just need to know what helices and sheets are and know that the way the entire protein folds up is tertiary structure. Secondary structure applies to parts of the chain folding up into helices and sheets. And then the way the entire thing folds up is tertiary structure. And then quaternary structure is what happens when you get several polypeptides, several chains of amino acids, each with its own tertiary structure, joining together. So your blood contains a protein called hemoglobin. Each molecule of hemoglobin is made of 4 chains of amino acids. Each one of those folds up into secondary and then tertiary structure, and then all 4 have to fit together to make a complete hemoglobin molecule. That's quaternary structure here. We have 4 chains of amino acids. Each one has its own primary, secondary, and tertiary structure. And then all 4 of them have to fit together to make a functioning molecule of hemoglobin. That's quaternary structure. This is a very important protein. I'm not going to have time to say much about it. It's called ATP synthetase. It is responsible for generating most of the energy that your body gets from the food that you take in. If you did not have this, you would die in seconds. You'd be completely unable to gain energy from your food. It happens, this is 2 different views of the same protein, and it happens to be made of 7 different polypeptides, each one of which is colored differently. You've got a chain of amino acids shown in red that makes a kind of central stalk, and then you have 6 chains of amino acids, each one of which takes the shape kind of a wedge. And then all 7 of them have to fit together so that you have these 6 wedges making up a kind of cap, and the 7th forming a central stalk. So you end up with a protein that looks kind of like a mushroom. And if you could not make all 7 of these chains of amino acids with amino acids in the right order, and then have them all fold up, and then have all 7 of them come together to make this, you would not be able to generate food from the energy, or generate energy, from the food that you take in. That's why this stuff is important. That's why I'm dragging you through it. I should mention that we've got researchers, or we did a few years ago in our computer science department, working on the problem of how can you predict the way that proteins are going to fold up into these shapes? And it turns out to be an extremely difficult problem in computation. We still can't do it yet. I don't think it's been cracked. It's very hard to predict just given the order of amino acids, what shape a protein is going to take. It's a lot harder than it looks. I actually had a student in here once who was working on this and was very excited by this part of the course because his professor had never really explained what he was doing. Well, now you know. And that's a very important problem in biotechnology for reasons I'll try to make clear later. One more example that you could actually taste. Some grains, notably wheat, contain proteins. This is one of them. It's called a gliadin. And it's a chain of amino acids in a particular order. That's the primary structure. Parts of this chain of amino acids have kinked up to form alpha helices, like the one I've circled, and others have kinked up to form beta sheets. That's secondary structure. The entire thing has folded up into this shape. That's tertiary structure. So that's a gliadin protein. There's other proteins in wheat called glutenin proteins. And collectively, gliadins and glutenins are collectively known as gluten. And there are some people that are sensitive to gluten, including my wife, for example. They can't eat food with gluten in it or it makes them sick. And this is what they're responding to. They have an immune sensitivity to these proteins that are found in wheat, to gliadins and glutenins. If you take some varieties of wheat have been bred by our old friend artificial selection to produce high protein, to produce lots of these gliadin and glutenin proteins. And if you take a variety of wheat that's been bred to have lots of these gliadin proteins and glutenin proteins, grind it, mix it with water, and then start kneading it, mashing it and stretching it and mashing it again, you make the proteins assemble into a giant quaternary structure that fills all of the dough. They assemble into this giant network, and that giant network not only fills up all of the dough, it holds together very tightly, and it makes the entire mass of dough very stretchy. So stretchy, in fact, that you can pick up the mass of dough and spin it around like this guy is doing. Proper pizza crust is made from varieties of wheat that have lots of protein in them. Incidentally, so is pasta. Pasta is made out of a wheat dough that's got lots of proteins that will link together as you're making the dough and then hold together tightly as you're pressing the dough into pasta shapes. So pasta and pizza crust are made from varieties of wheat that have lots of these proteins. And as you knead the dough, the proteins assemble into a huge quaternary structure that makes the whole dough hold together. That's how this guy can get away with spinning that pizza dough. That's what keeps the dough from just breaking up into blobs that go flying everywhere. Speaking of which, if you take a variety of wheat that's got low protein, you don't get a dough that holds together because it's got much less glutenin and gliadin. So you don't make that giant quaternary structure. Instead of making a dough, you make a batter with those. And if you bake baked goods with low protein varieties of wheat, because you don't get that giant quaternary structure, the end result is not chewy, it's crumbly and delicate. This is why with cakes and pastries and pie crusts, you need low protein varieties of wheat to make those. And you'll end up with a product that is, well, cake textured, that's crumbly, not chewy. This is why you can't pick up a massive cake batter and spin it around the way you can a pizza crust. It's all got to do with different amounts of protein in the wheat. A lot of the flour in your grocery store is called all-purpose flour, and that's kind of a compromise between these. You can use it for a lot of things, but if you're a serious baker, for a lot of your creations, you probably get flour specifically made for cake. You'd get cake flour. Or if you're serious about making pizza, you'd probably go out of your way to buy pizza flour, all of which you can also get in supermarkets. And the difference is the amounts of protein that they've got. And if you can't eat gluten at all, you have to replace the gluten with non-gluten proteins that will make your product stick together. Egg can be used, whey, proteins derived from legumes like beans, for example. Those can imitate the effects of gluten to an extent. Pomi, please mute. Please mute. Please mute. All right, I'm done. Gluten-free bread tends to come out more crumbly because it doesn't have high amounts of gluten. All right, we're done. I'm going to stop the recording.